DNA structure is helical in nature, and its backbone is connected by phosphodiester bonds, and the linkages in the middle are nitrogenous bases, and this connects both backbones of DNA. Within each unit of DNA is a nucleotide, and it contains a phosphate which has two negative oxygens, which gives DNA an overall negative charge naturally, and it has a nitrogenous base and a 5-carbon sugar. The significance of the 5'-carbon sugar is that at the 1' position, there will always be a nitrogenous base connected in this fashion, and at the 2' carbon, there will be an OH for DNA. This determines whether you're seeing DNA or RNA. In RNA, you won't have a OH, you just have an H. At 3, this is where DNA is connected throughout its backbone to another phosphate group. And at the 5' carbon, this is where DNA will always add another phosphate group, growing its chain. The 5' carbon explains the anti-parallel nature of DNA. You always see a 3' to 5' backbone and another backbone that is opposite of that backbone, which is 3' to 5'. DNA structures form major and minor grooves because of their coiling in certain directions. At the minor groove, there's going to be less protein interactions because there's less surface area. And in the major grooves, there would be more protein interactions. Also, the coiling in certain directions for a certain DNA will determine its handedness. This DNA right here, since you see it's rising to the right, it's right-handed. And this DNA strand is left-handed because it's rising to the left. Nitrogenous bases connect DNA strands together by forming hydrogen bonds between each other. And there are two types of nitrogenous bases. There are purines, which have 2 rings, and pyrimidines, which have 1 ring. They arrange themselves almost stacking on top of each other because of their hydrophobic nature, because DNA strands are usually in presence of high amounts of water. In DNA, bases pair based upon adenine to thymine and cytosine to guanine. I remember this by remembering apples are in the trees, apples are adenine, thymine's tree, and cars are in your garage, so car is cytosine and garage is guanine. What happened to the melting temperature of DNA structure if it were exposed to a higher concentration of a saline sodium citrate buffer? The backbone of DNA is negatively charged because of the presence of phosphate ions, and saline sodium citrate has a positive sodium that's going to bind to the negative backbone. This will therefore strengthen the structure of DNA and cause an increase needed for the melting temperature. Think of it almost as 2 magnets that need a positive and negative side to bind. This DNA strand, when compared to others that aren't in the same concentration of saline, will need a higher temperature to denature it instead of a lower one.